So let's take a more in-depth look at what happens during transcription. So we know that RNA is made from DNA, and we know that it occurs in the nucleus because that is where our DNA is stuck. It is stuck in the nucleus of eukaryotic cells. So this image here is showing you how transcription happens in eukaryotes. So these two strands right here are showing you the double-stranded DNA. So this is the kind of like the base molecule. Remember that this is the gene, all right? And this gene is going to be providing the instructions ultimately for how to make a protein. But before we can make a protein, we first have to make mRNA. So what happens is that the gene is actually down here, kind of like downstream of this area that we call the promoter. And what happens in this promoter region is that we have transcription factors, which are proteins, and we have enzymes, and we have some other stuff that comes together and it binds to the DNA at this promoter region. And you can see that we call the promoter region, we kind of call it this ta-ta box. And it's because it's made up of a whole bunch of thymines and adenines that are repeating in this area. And so there's some sort of signal to the transcription factors that is where they should initially bind to and then start the transcription process. And as you can see here, we've got our double-stranded DNA, and we have just one small area of the DNA that unwinds. So we're not completely separating the two strands of DNA like we did during DNA replication. Instead, we're just opening up just the gene and a little bit of the area around the gene. So we're really just exposing the gene that we want to transcribe. And we call this the transcription bubble because it's only this area that's going to be transcribed into RNA. So the two strands of DNA separate and the DNA bases become exposed. Now only one strand is going to get transcribed into forming this mRNA molecule. And so there is an enzyme, RNA polymerase, and if you look at the word RNA polymerase, it tells you what it does. Remember that ACE, that ending of ASE, means that it's an enzyme. And it's an enzyme that makes a polymer of RNA. So it's taking those nucleotide monomers and putting them together in order to form a polymer, which is a longer, bigger strand of RNA. Once the gene is transcribed completely, the mRNA is going to detach from the area, and the two strands of the DNA are actually going to come back together as if nothing happened. So we're not damaging or destroying DNA in the process, we're just temporarily opening it up so that we can make this molecule of mRNA, and then it goes back together until that gene needs to be transcribed again. Eukaryotic DNA actually contains, or the genes contain segments of DNA that are needed to make the protein, but there are also some kind of areas of DNA that are not needed to make the protein. And in fact, they kind of get in the way when trying to make a protein. So exons are just these areas of genetic sequences or DNA that we need in order to code for the protein. So on the picture down here, we're looking at these blue segments here. Now the orange segments are indicating introns, and introns are just sections of DNA that actually get in the way. They don't code for a protein. I don't know if we know really what they do. They might have something to do with which genes are expressed, or we might just not know. But essentially, before we go into our next step of translation, we have to get rid of these areas of genetic information that need to not be there. Now, remember that in DNA, the mRNA is being made from the DNA. So these nucleotides, so let's say we've got some C's and G's and T's and A's and all that in here, they're still getting converted into our mRNA. So what we end up with is a strand of mRNA that has some useful information, but also some information that we need to get rid of before we go into our next step of translation. So after transcription happens, so we can see here that the DNA gets converted into the mRNA. But this mRNA needs to be processed because it contains the introns and the exons. And we have to get rid of those introns because remember, the introns they're not needed to make proteins, and in fact, they're going to get in the way. So what happens during this post-transcriptional processing is that after transcription happens, there are enzymes that go in and they remove these nonsensical areas that are not needed. And then it actually splices the RNA back together. So we end up with this mRNA molecule that has the correct information needed to make a gene, but we've gotten rid of all of the information that we don't need. So we end up with this kind of shortened RNA molecule that only contains the information that we need. Now, in addition to cutting out the introns in order to only have the exons, the ends of the RNA molecules also get some extra nucleotides added to them. So you can kind of see that on this picture here. You get a 5' cap, and so these are just extra nucleotides added to the 5' end of the mRNA molecule. At the other end, you get this thing called a poly A tail, which is just a whole bunch of adenines added on to that end of the mRNA as well. And these added nucleotides primarily serve a protective function. So think about it, this molecule is going to have to go from the nucleus, and it's going to have to squeeze out through those nuclear pores in order to get to the cytoplasm. And along the way, it might bump into things, it might be exposed to some chemicals that could be dangerous to it. And so these extra nucleotides are sacrificial, because if we lose a little bit of information here, it's no big deal because we don't need this information in order to make a protein. Whereas if we lose information from the actual coding part of the mRNA, now we're going to end up with something like a mutation, and we're going to end up with a protein that isn't made properly. Now we're going to take a look at the second part of protein synthesis. So during transcription, we made a molecule of RNA, and more specifically, we made a molecule of mRNA. Now the mRNA, remember the DNA is stuck in the nucleus, but to make a protein, we need an organelle called a ribosome. And so the mRNA is able to carry the information in the DNA in the nucleus out to the ribosome, which is located in the cytoplasm.